Hey guys, this is Mrs. Hunt. I'm sorry I'm absent today. My son has an ear infection and I had to be out. So I hope you guys are able to get all your work done today and email me or text me on remind if you have any questions. Okay, so what I've done for you is I've made my notes from the board. So this is what I would normally have done had I been in the classroom. I'm going to run through this with you guys. Okay, so you're going to pause the video now and draw these on your paper. This is what you're going to see looking at microscopes. So first of all, you see a bacteria cell here, and you know that it's very, very small, and that you have cilia hanging off the outside. You have a cell wall on the outside, and you have a plasma membrane on the inside. Plasma membrane is also called the cell membrane, so you have to memorize both. Coming out the end is flagella that helps it move. On the inside, you have ribosomes, and you can have circular DNA, but you won't see any of the insides using our microscopes. Bacteria cells are the smallest, and they come in three shapes. So there's coccus, which are circular shape, bacillus, which are a rod or pill-like shape, and spiralia, which are spiral shape. When you go to check out your microscope slides today, they'll have those three shapes on them. All right, a plant cell's general shape is square. The cell wall is on the outside. The plasma membrane is on the inside. The cytoplasm is the fluid inside. These are eukaryotes. They have a nucleus and membrane-bound organelles, but you can't see the organelles because they're too small. It's going to have a big vacuole that you might be able to see by a faint line inside here. You might see a few ribosomes. The plant cells are the biggest. Then we get to an animal cell. The animal cell is the plasma membrane on the outside. Remember, they don't have a cell wall. And they're also eukaryotes. We have a nucleus. You might find a few ribosomes and then the cytoplasm. And the animal cells are more medium size. Okay? So we talk in class about why cells are so small. And we talk about why and what needs to move through a cell. So we introduced this word diffusion, which means movement of particles through a membrane. Diffusion is going to be the movement of these molecules in and out of a cell. So here is coming in, we have food. Remember, characteristics of life, having a metabolism is one of those things we talked about. So we need to take in food as protein, lipids, which are fat, carbohydrates, which are sugar, nucleic acids, and we memorize the elements of life as carbon, hydrogen, nitrogen, oxygen, and phosphorus. It spells or makes Schnapps sound, so that's kind of easy for us to remember. Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. All right, I also said that carbohydrates are a sugar. Glucose is one of those sugars. Sugars often end with oses, so glucose, fructose, maltose, dextrose, lactose are all sugars. And I wrote the formula for a simple sugar up here. Okay? Water comes in, oxygen comes in to our cell, and then out comes CO2, water, and salt, NaCl, are some of the salts. So these are all things that need to move in and out of our cell. Okay? If you want to pause the video here, you can, and draw this down on your paper. One of the slides you're going to see in the notes section is about the surface to volume ratio. So here are surface areas of cubes. So remember, surface area is length times width times number of sides. So here we have a surface area of 2. So this is saying 2 times 6, number of sides, is 24. The volume is length times width times height. So here our volume of our cell is 2 times 2 times 2, which is 8. And then a ratio is basically comparing one to the other. So we're comparing that surface to the volume. When we divide it, we get a big number of 3. Let's take a look at a bigger cell. Here, our next size up, our surface area would be 4 times 4 times 6, which is 96. The volume, 4 times 4 times 4, is 64. I've gone up 2 sizes. I've doubled my size, but my surface area is not doubled, and my volume has definitely gone up a lot more. When I divide the surface area by my volume, I see that the ratio is 1.5. It looks like the ratio is going down. It's getting smaller as we go up. All right, let's look at this bigger one. Surface area of 216, volume of 216, and we're at a 1-to-1 ratio. And as we get really big, 284 is my surface area, but whoa, our volume is bigger than our surface area. So you're thinking, what does this mean? I don't get it. All right, let's go back here. I want you to think of a plasma membrane as a door that lets these things move in and out, these nutrients move in and out. Now let's go back here. What if I said that there was a room and the room has 24 doors? And in those 24-doored room, only 8 things need to move in and out. This starts to be very easy. And it gets let's call it a speed of 3. It moves so quick that everything can exit in and out very quickly. We have a ratio of a whole number of 3. We get to a bigger cell, we have 96 doors. Now that's a lot of doors. But the volume is 64. 64 things need to move in and out. That's a lot. And it slows down. So we're at a 1.5 on our surface to volume ratio. This is starting to slow down. We get a bigger cube, 216 doors, 216 things that need to move in and out. We're at a surface area ratio of 1. All right, look at our big cell. 384 centimeters cubed, 384 doors with a volume of 512 things that need to move in and out. This is going to be really slow. So you start to see that the surface area to volume is a lot slower. So let's clarify. In a small cell, the surface area to volume is bigger and everything's able to move quicker. In a big cell, the surface to volume ratio is small and things don't diffuse as quickly. In class, I would have done a diffusion demonstration for you all, and I'm sorry. Diffusion is the movement of substances through a cell membrane, and we're going to use agar cubes and vinegar to move through the agar cubes. The agar has a dye in it called Blumenthal blue, and when it gets exposed to an acid, which the vinegar is an acid, the cube changes colors. By the way, is chemical or physical change when something changes colors? If you guessed a chemical change, then you're correct. All right, which tube will diffuse faster? Which cube has the larger surface to volume ratio? All right, so here are the 3 cups, and here are the 3 agar cubes, small, medium, and large. Which one will go first? We take the cubes and we put them in the vinegar. And look, you see how they start to change color. This is the agar, or the Blumenthal blue, reacting to the acid of the vinegar. But let's think about it as the vinegar is diffusing into the cells. See how the small one is almost all the way diffused? And then we have our bigger cells. After they were all the way, the small one was all the way, we took them out. And then we cut them open. The small cube had already diffused everything through the cell. The medium cube was slower. And of course, the biggest cube still had the most to diffuse. It was the slowest. So the small cube had a big surface to volume ratio. That let it go by quickly. The big cube had a small surface to volume ratio. And that made it go really slow. We timed it, and it was 14 minutes to go through the small one. I let the big ones go and just to see, and I got almost 2 hours. Can you imagine if this was a breath of oxygen, how long it would take to get all the way through that cell? Okay, now that we've done that, okay, the band is going to start to play. Now that you've gone through these and you've seen the diffusion lab and you've drawn pictures from the board, now it's time to do some drawings. I've got up front on the lab table a bacteria cell, plant cells, and animal cells. And you guys have slides already ready to go. I want you to copy this on your paper, a bacteria cell, a plant cell, and an animal cell. I want you to, I'm going to move my face here, write down the magnification that you're going to observe these 3 things under. Notes of when you draw from a microscope. Make sure that you are looking at the image correctly. We just drew what you should see. So if you say this is a plant cell, right here, I'll say you drew a bunch of dirt. So that was not very clear. Make sure you're seeing what you're supposed to see. Take some time. Go slow on this. All right. Also, draw what you see. Don't checkerboard it. Don't rush through. Make sure you give me all the good details. A scientist makes a good observation. And finally, at the bottom here, it says, record the magnification. Remember, if you're on the red objective lens, that's a 4, and that's a 40 times power. If you're on the yellow, you're at 100 times power. And if you're on the blue, you're at 400 times power. Make sure you record the strength, okay? I put this on here just in case you were having a really hard time finding something, then you can use these pictures. This is Coccus bacteria at 400 times magnification. You don't see much of the details, but you can see its circular shape. This is a plant, an onion root tip at 400. And this is a human cheek cell. Can you find the nucleus? Right there it is. Okay. So you can use these pictures to be able to use if you can't find them under the microscope. Okay. After you finish this, then you're going to go back into the classroom. I want you to look and make sure you've got everything turned in for Module 2. Make sure everything is turned in. And then you can begin Module 3. Module 3, you have an introduction to the cell. You have a Nearpod to do. You have reading with no notes. There are textbooks by the green shelf in my classroom you can use. There's some videos to watch on here. There's a Jamboard to do. And then you have a quiz and another quiz to be able to take. So hopefully my absence won't create too much of a block in everything that's happening. And you're able to get your work done, and you guys stay healthy and have a great weekend. All right. Thanks, guys.